Hello students, welcome back to the channel. So in this video we are going to study about the complex permanent tissues. Now see, complex permanent tissues are known as complex. Reason is they are made up of more than one type of cell. Okay, more than one type of cell. This is important. And because they are made up of more than one type of cell, we call these tissues as heterogeneous tissue also. Is it clear? Now, what are the two types of the complex permanent tissue? One is your xylem, second is phloem. Xylem is responsible for conduction of water and mineral, and phloem is responsible for the conduction of the food. That is why these complex permanent tissues, they are also known as conducting tissues or vascular tissue. They are also known as conducting tissue or vascular tissue. Reason is clear, because they are helping in conduction. And vascular, why? Because in our body, in a human body, if you know, blood is a vascular tissue which is responsible for the conduction of the food as well as for the gaseous exchange and transport of the gases from one cell to another cell this way. So that is why it is also known as vascular tissue. Now see, complex tissue means xylem and phloem, they are absent in gametophyte. We have already studied in the plant kingdom. The plant kingdom is having two types of body. One is a sporophyte body, one is gametophyte body. So gametophyte body does not have any complex tissue, means they don't have xylem or phloem. That is why we say in entire plant kingdom, gametophyte is atracheophyte. Okay, in entire plant kingdom, gametophyte is a tracheophyte, means they don't have vascular tissue. So I hope this term is clear to you, and it's very important from the competitive point. Is it clear? Now let us study xylem in detail in this video. So in this video, we are going to study about the xylem in detail. Now this term xylem was given by Nageli. And the function of the xylem is for the conduction of water and mineral. So if they have to do the conduction of water and mineral, then obviously the cell need to be dead so that their working efficiency can be increased. So cells are dead cells. Only one component will be living, but majority will be the dead cells. If I'm saying dead cells, means protoplast is not there. Protoplast is absent. Instead of protoplast, what is present? Lumen is present there. So for the efficient conduction of water, death of protoplasm is must. Is it clear? Because dead tissues are more developed in the water scarce conditions. They can do the conduction very well. Now, in hydrophytes, those plants which are found in the aquatic habitats are known as hydrophytes. So in hydrophytes, xylem is poorly developed. Is not well developed, okay? It is poorly developed. Reason is that they don't require the xylem for the conduction because they are already present in the water. So they can take the water by the process of simple diffusion. But in xerophyte means dry condition, like desert plants. Xylem is well developed. So in hydrophytes, it is poorly developed, and in the xerophyte, it is well developed. Is it clear? Now, on the basis of origin, xylem is divided into two, primary xylem and secondary xylem. And secondary xylem. Clear? Now, primary xylem is the one which is developed from procambium during vascularization. It developed from procambium. and it is developing at the time when vascularization was going on. But secondary xylem is developing from vascular cambium. It developed from vascular cambium. And it is developing at the time of the secondary growth. So first of all, this is clear that when their origin is there, at what time their origin is going on. Second, if you see here, the primary xylem is known as xylem which is formed early in the life. It is formed early in the life of the plant. And this is developing when? At the time of secondary growth. Now, on the basis of development, primary xylem is again divided into two. On the basis of development, primary xylem is divided into two. One is your protoxylem, next is your metaxylem. Protoxylem, okay, in which lignification will be started before the completion of elongation. So lignification occur before elongation complete. But in case of metaxylem, lignification will occur after elongation complete. Lignification occur after elongation complete. But such terminologies we will not use with the secondary xylem. The secondary xylem, we will not say it is having protoxylem and metaxylem. And this is very, very important, very important. Protoxylem and metaxylem, they both are absent in secondary xylem. So protoxylem and metaxylem, this term we use only with respect to the primary xylem and not with respect to secondary xylem. So crossword means they are absent. Is it clear? I hope this much topic is clear to you. Now, next. Now see the difference between the protoxylem and metaxylem. This is the first formed primary xylem. First formed primary xylem. And later formed primary xylem is known as metaxylem. Is it clear? Another difference you can find with respect to the different types of thickening which are present. See here, let us assume this is your protoxylem. So here you are finding the thickening this way. Lignin deposition is occurring this way, in this pattern. Or lignin deposition can occur in this pattern also. But here lignin deposition pattern will be this way. Or it can be in this manner also. Now see, this is your annular. First is annular. This is your spiral thickening of the lignin. This is reticulate thickening. And this is simple pit. And this is bordered pit. Is it clear? So protoxylem are having the lignin deposition as annular and spiral. And metaxylem is having its lignin deposition as reticulated and pitted. Now see the learning tips. For here you can remember MRP. Metaxylem will have reticulated and pitted thickening. And for here you can remember pars. Protoxylem is having annular and spiral. Only this is just, I've written it with no meaning. It is not having any meaning, okay? Only just for making the pars, I have to write this word pars. So protoxylem has annular and spiral, and metaxylem is having reticulated and pitted thickening with respect to the xylem. This is all what we have shown it. It is actually how the lignin deposition is going on, okay? Lignin deposition. In both the cases, how the lignin deposition is going on. This only we have explained through diagram. Now next classification is on the basis of protoxylem. Now see, this is your young formed xylem, newly formed, and slowly it is maturing. You can check the board carefully. Maturation is moving in the center. Okay, let us assume this is the periphery part. Let me draw the circle. This is the periphery part and maturation is coming in the center. Okay, so this is your primary xylem, which is a type of protoxylem, and this is your metaxylem. So if you see in the xylem, in the diagram, protoxylem is towards periphery. It is towards periphery. And when protoxylem is towards periphery, then this condition is known as exarch. Is it clear? And if you see, metaxylem is towards center. It is towards center. So we will say this condition is known as centripetal. So we will say exarch xylem is having centripetal growth. Because if you see continuously growth is going towards the center. Check the growth is coming towards the center. Understood why I'm drawing it double? Because lignin deposition is there. As a lignin deposition, the cell is becoming thick. So when you study the TS, you will find it is having two layer. That is why I'm drawing it two layer. Understood? So basically you have to just remember that protoxylem is present where. If it is present outside, you call it as exarch. And you have to remember if protoxylem is outside, means metaxylem will be towards center. It means slowly meta formation is going towards center to centripetal growth. So simple you can understand exarch xylem shows centripetal growth. Is it clear? So one type of classification is exarch xylem. Now see in the diagram endarch. Now see here, this is in the C, check the board carefully. This is your protoxylem I'm drawing. And slowly the maturation is moving away from the center. Understanding the diagram? Maturation is moving away from the center. So this is your protoxylem. And this is your metaxylem. Now I hope you can tell me where the protoxylem is present towards center and metaxylem is towards periphery. So when protoxylem is towards center, I will say this is endarch condition, means inside, endarch. And metaxylem towards periphery, I will say centrifugal. Is it clear? So this is your second type of xylem classification with respect to the position of protoxylem. I hope this concept is getting clear. And we will say endarch xylem. Endarch xylem shows centrifugal growth. Centrifugal growth. Another important concept, this condition, exarch condition, is seen in the root anatomy. This is seen in the root anatomy, and this condition will be seen in the stem anatomy. So with respect to the position of the protoxylem, I hope the concept is clear to you. Now let us discuss the component of the xylems. Now see here, xylem is having four elements. Tracheas, tracheae also known as vessels, xylem fiber and xylem parenchyma. This together, tracheates and tracheae together are known as trachea element. This is again very important PMT question. Trachea elements. Is it clear? So these are the one things. Now see here in this, these three components are dead, and they are dead due to lignin deposition. And only living component is the xylem parenchyma. This is the only one living component of the xylem. Is it clear? Now let us study the tracheates and trachees. Now see the difference between them. Tracheates and tracheae means vessels. They are elongated tube-like cell. Very important line of NCERT. They are tube-like cell, and here we say they are elongated tube-like structure. Tube-like structure. This is very, very important. Check in your line of NCERT, you will find it. One is tube-like cell and one is tube-like structure. I hope this concept is getting clear. Is it clear? Now see one thing. Try to understand the diagram here. This is your one xylem fiber. Okay. This is the one xylem fiber which is having highly lignified wall. Let me draw. This is a lignin deposition is going on. So because of the lignin deposition, you know very well the cell has become dead. With the lumen is also present. With the very narrow lumen will be there, and these are the pits present. These are the pits. So what the nature has done, nature has kept the many xylem fiber in such a way that pit pair should come in contact. The pit pair should come in contact. You can see here. Okay, let me draw the third fiber. Then this way the next fiber. So pit pair is coming in contact, and this is now how the tracheates are made. This is how the nature is making the tracheates by using the fiber. So right now, this is only for learning tips. When I'm saying fiber, I'm using, it means fiber, you know very well, it's dead. The tracheates are also dead. And dead because of the deposition of the lignin. And now see pit pair are made in the contact so that water can pass. Check the water movement. It is passing this way. Is it clear? And if you see individual cell is maintaining its identity. Individual cell, let us take this cell. So this cell is also maintaining its identity. And if you see it is a pointed at both ends, this is pointed. So we say elongated tube-like cells which are pointed at both the ends, and they are having the pit pair in connection. So they are having the pits formation is going on, which is already started in the sclerenchyma fiber. So pit formation is there, but they are not showing the simple pits. They are showing which type of pits? They show bordered pits. And for this, you can go and check the sclerenchyma fibers video, then the topic will be clear. What are bordered pits? Is it clear? So they are having the bordered pits. And tracheates are the one which are seen in. Now see, they are seen in pteridophyte. They are seen in the gymnosperms, and they are also present in the angiosperms. But in angiosperms, they will not do much conduction for the water, and they are just providing the support, working as a mechanical tissue, just like sclerenchyma fiber. And that is why in case of the angiosperms, we say the tracheates are working as a fiber, and the tracheates are known as fiber tracheates. In case of angiosperms, tracheates are working like a fiber, and we call them fiber tracheates. Because the conduction of the work in angiosperm is given to vessels. So vessels are seen in angiosperms. Now try to understand. See, this is my one fiber. Let us assume this is one fiber. Okay. I'm placing now end to end. I'm not placing on side to side. I'm placing end to end. This is the way I'm placing end to end. Try to understand the diagram. The topic will be easy for you. Okay. And this is the lignin deposition. Let me do the lignin deposition also. highly lignified they are, and fiber are with the pits only, pits will be there. Pits will be there, so let me draw the pits also. See here, these are the pits. But one more additional thing will be there. You have to check the board carefully. Wait, what I will do, check. This common wall is getting dissolved. The common wall which is holding them, that is getting dissolved. That is why they are tube-like structure. They are not cell because they have lost their individuality. And the many cells are participating. So each is known as vessel member. And tell me one thing, isn't it more efficient in sending the water up? Because the water will go straight without any hurdle. No hurdle. And I'll say they have created what? What they were creating this area when they were dissolving. They have created perforation. They have created perforation plate. This is again very important PMT question ki perforation plates are present in. They are present in tracheae, not in tracheids. Tracheids will have pits, but tracheae means vessels will have perforation plates, plus they will have pits also. They are having pits also. These are the pits also present. Bordered pits. Is it clear? So based on this question was asked, कि trachea elements, and I told you trachea elements means tracheids and vessels. Trachea element differ from another trachea. How? The trachea elements means tracheids and vessels, they are differing from tracheids in being perforate. It means they are having perforation plate. You check in your NCRT also, NCRT has used the word perforation. They have only used the word perforation. Is it clear? So it means here I will say in the tracheids it is imperforate. They are imperforate because they don't have perforation plate. But vessels are the one which are having perforation plates. So I hope the difference between the two is clear to you. It is very, very, very important concept. Is it clear? So can we say that tracheids are unicellular? Because individual cell is maintaining their, because they are maintaining tube-like cells. So can I say it is unicellular? Because individual cell is maintaining its identity. But here, if you see, it is multicellular. And multicellular, all the cells are dead. So we will call vessel is an example of dead syncyte. Syncyte means fusion of cell, but it's a fusion of dead cell. So it's a fusion of dead cell. Is it clear? An individual member, this one member, this way, one member is known as vessel member. Individual is known as vessel member. Again, it's an important line of NCRT. Is it clear? So now let me give you another concept. Now see, vessels are characteristic feature of angiosperm. This is again very important line of NCRT. They are the characteristic feature of the angiosperm. But now I'm giving you few points with respect to the competitive areas. Okay. So I'm just writing it as an additional point, few points for the competitive points. Try to understand it. See here, few gymnosperms also possess vessels. Exceptions now. Gymnosperms, these are exception area, okay? Gymnosperms with vessels. And examples are your order Guinea-tails, and this order Guinea-tails include Guineatum, Ephedra, and Welwitschia. Is it clear? Few pteridophytes are also with vessels. Example is Selaginella, Equisetum, and Pteridium. Is it clear? Now, few angiosperms families don't have vessels. Next is vessels are absent in few angiosperms family. Angiosperms family which lacks vessels. So these families are Trachea, Trochodendrocea, Tetracentracea. These are the families which does not have any vessels. And now few angiosperms examples which does not have vessels. Angiosperms which lack vessels. For example, your Dracena, Yucca, Tegenaria, Trimis. These are the one which does not have vessels. Is it clear? Now see, we have to discuss about the xylem fiber. I'm discussing about xylem fiber, which is also known as wood fiber. Okay. This is the third component of the xylem, which I'm discussing right now. And it will be more abundant in the secondary xylem. It will be more abundant in secondary xylem. Okay. And main thing is this, they are also highly lignified, and so much lignification is going on that they have obliterated central lumen. So they are highly lignified, and because of high lignification, they have obliterated lumen. This is again question area. So obliterated lumen is present where? It is present in the xylem fiber. Is it clear? They are not found in the gymnosperm wood. They are absent in gymnosperm wood. And because they are absent in gymnosperm wood, that is why the gymnosperm wood is also known as softwood. Gymnosperm wood is known as softwood. Is it clear? Now next component left is your xylem parenchyma. And xylem parenchyma is the only living component of the xylem which is responsible for the radial conduction of the food. As it is doing the radial conduction of the food, it is also known as ray parenchyma. The ray parenchyma is responsible for radial conduction of food. Radial conduction. And they are the one who will store the food also. Okay. The storage is done. This is again important. Storage food is either starch or fat, and another component, other substance is tannin. So I hope that in today's video, we have discussed about the complex permanent tissues, which are called complex because they are heterogeneous. Two types are there, xylem and phloem. And xylem is again divided into primary xylem, secondary xylem. Primary xylem is again divided into protoxylem and metaxylem. And based on the position of protoxylem, we have studied exarch xylem and dark xylem. Okay. This is the classification we have studied, and we have studied in detail what are the components of the xylem. So total 4 components are there: tracheids, vessels, xylem fiber, and xylem parenchyma. Now, if suppose I ask you a question, who is tube-like cell? Your answer will be tracheids. And who is tube-like structure? Your answer will be vessels. So I hope this explanation topic is clear to you. Thanks for watching.